Hi, everyone. Thanks for the opportunity to speak here. So this presentation is based on that one, and I tried to move it 100% to the notebook, and some things aren't working. So it won't be live, but at least you'll see some of the capabilities. So this is using Damian Avila's live reveal.js. So you get a presentation that can also execute code. So that ran a line of code even though you didn't see it. Does that goes off the page. Maybe I'll zoom out. Oh, that makes sense. Sorry, 1 second. All right. Okay, so that's the title of my talk, Clustering of High-Content Screen Images to Discover Off-Target Phenotypes. So I'll tell you what all of that is about first. And yeah, as I said, this notebook has the entire analysis, but some of the stuff isn't working, so it won't be live. Okay, so what is high-content screening and why do you care about it? So the first question is reverse genetics on a massive scale. So for those of you that don't do biology, forward genetics is sort of the traditional way to do genetics, and you go from a mutation, so an interesting trait, a phenotype. For example, in the fruit fly, one of the classic ones is curly wings. So some flies have curly wings and other flies have straight wings. So you go from that, and then using breeding and various other approaches, you find out which gene is responsible for that trait. Okay, so that's reverse genetics. That's forward genetics. Reverse genetics is when you mutate a specific gene and you observe what happens to the organism. So that's why it's in the other direction. And in high-content screening, what you're doing is on each of these wells in a 384 well plate, you put some cells, and then every well gets a different treatment. Usually you're disrupting a specific gene, and then you take a picture. And you do this for, say, 50,000, 100,000, 500,000 images. So you're testing the effect of all of these disruptions at once on the shape of your cells. So that's high-content screening. By the way, the talk's a bit short, so feel free to ask questions throughout it. Why should you care about it? Because in the past decade, or in the aughts rather, from this study, more new drugs were discovered from high-content screening than from, say, molecular modeling-based approaches, even though the latter actually had way more resources thrown at it. So it's actually a very effective way to find drugs, treat diseases, and just generally do biological discovery. Okay, and why do I want to do clustering? And that's because I find that the traditional way to analyze high-content screening is pretty limiting. So this is just from a figure from a high-content screening paper. The main point, there's no laser pointer, I'll use my mouse. The main point is this panel. So these two axes are essentially replicates of one another. That's why it's highly correlated. So all you're doing is taking one variable. I don't really know what it is here, something like cell density. And you're ranking all of your images across that one variable, and then you look at the low-ranking images, the high-ranking images. But obviously, there can be a lot more going on in your pictures. So cells can take all forms of shapes, independent of the density. So essentially, you're spending $250,000 on a screen, and then you're getting this one number out of it, maybe a list of 100 genes that look interesting, and that's it. And as I said, it's actually very valuable. Great drugs. There's been a lot of success in drug discovery, but there's a lot more information in that data that people aren't using. And clustering is a nice way to discover that, because if you have one image where your cells look weird in a particular way, then that doesn't necessarily tell you much, because in biology, stuff goes wrong all the time, and you don't know whether that thing was a fluke or if it's a real effect. Whereas if you get a cluster of images that all look that way, then you might get a clue that you're onto something. Okay. So again, this is a notebook demo, so I can run this. And again, we're not actually going to run this, but I just want to emphasize that you can do this live. Okay, so the first step, if you're familiar with clustering, you want to describe every image as a set of features. So that's the first step, is to map your images to feature or descriptors, they're sometimes called. So just some way to summarize the image. And so first thing we're going to do is write the map function. So this is from the screen that I've been mostly working on. It's from Christophe Marcel at Monash. And the main thing that I want you to take away from this image is that there is no way you're going to count cells or find the cells in here. These are muscle precursor cells, and what they do is they start fusing together when you put certain factors into the thing in order to form, muscle fibers. And so we're interested in things that promote muscle formation or muscle fiber formation or disrupt it. But the point is, once they start fusing and getting all these spindly shapes, you're not going to find where the cells are. And so designing features for this is challenging. And there's this great quote from the speech recognition field, which I took to heart, and it's from Fred Jelinek. Every time I fire a linguist, the performance of our speech recognition system goes up. So the moral of the story from this, it might be apocryphal, but I think it's really good, is that sometimes machines and doing sort of random okay things does as well as intuition or better. And you don't need the precise knowledge, you just need to get some information out of your data, statistical information. So knowing this, I plowed bravely on and I did some feature mapping. So this is my main function, feature map, take an image and sample size, and I'll tell you what that's about in a second. So I call this don't care feature extraction. So I don't care that I'm not finding the cells. So all I'm doing here is I'm thresholding, very simple threshold, and then I'm doing connected components, so things that are isolated from each other. So you can see, obviously, none of these objects are individual cells, or maybe some of them are, but mostly they're agglomerations of cells of various forms. And we're just going to measure properties on those. And because the data had some illumination issues in some cases, so I'll scroll that. So you can see here, like, the center of the image is a bit less illuminated than the outside. I use two different thresholdings, and I measure the objects in both. And I just concatenate all the features together. So very, very simple. What are the features? I'll skip this bit because it's very long, but this one here is where you want to see it. So for every object in that image, I'm measuring these properties using scikit-image. So the area, how big that object is, the eccentricity, how elongated versus spherical it is. Euler number is whether it has holes in it or it's one solid block. Extent is how big it is. Mean, max, and mean intensity, and solidity, which is how much of a sphere it fills up. So I measure that for all of the objects, and then, or some subsample of the objects, and then I take the quantiles, 5th, 25th, and that summarizes my image. Okay, and this is the thing that's not working, so I'll skip over it. So find all the images. Scandir, you should check it out. It's a way to recursively walk your directories, and it's a lot faster than the built-in os.walk. And then we do the conversion, and we put it into a pandas data frame, which you all know about now. So this gives me a 2D matrix of floats, and the index is just the coordinates, which is the plate ID and the coordinates on that plate, and the columns are the feature names. And so what does that look like? It looks like that, right? So the number of objects in the image in this particular image is 481. The fifth percentile of object size is 13 pixels. 25th percentile is 20 pixels, and so on. Okay? So again, these are very, very unsupervised features. MongoDB is what I use to keep track of these relationships. So every image corresponds to a gene that has been disrupted. And that corresponds to an image file somewhere on my disk or in a server somewhere. And server-side processing is one of the things that I haven't done yet, but would like to. And then the coordinates and the wells. And there's obviously other information that you can gather. So for those of you that don't know, MongoDB is like the database for people who don't know or want to deal with databases. And a single entry in a MongoDB is like a JSON file. It's just a bunch of key value collections. And it's fast, open source, has a lot of support, and it has Python bindings. So highly recommended. And so we can start a daemon, which I'm not. Okay, so that's starting the MongoDB process in my computer. And just so you know what it is, you get a client to the daemon, and then you give it the database name and the collection name. And you get that. And hopefully, and if you do a search, this is what a document looks like. Okay, so this is the ID of that document, and yeah, you've got the barcode, the file name, etc. So it's super easy. Then we use scikit-learn to do clustering. And yeah, all I'm doing is scaling the data so that everything has 0 mean and 2 variance, so that all the features are on the same scale, because as you saw, my features are all over the place. And then we do mini batch k-means, which is a scalable version of k-means, to cluster all the images. So let's have a look at what that looks like. So this is just some setup code to get the genes from the cluster ID. And these are all the cluster sizes. So one of the things to take away is I'm not super happy with k-means, because you get this very heterogeneous cluster size, or at least with my data. So yeah. And so pick a couple of clusters. And so these are all the genes in one of the clusters. And I'll show you what the images look like. All right. So this is a set of 6 images in one cluster. You don't have a baseline for how similar they are, but they do have similar features, right? And if you look at another cluster, you can see that they're completely different to that previous one and quite similar to each other. Right? So that is the main message from this talk, is that you can do something really simple, borderline stupid with your data, and still get useful results if you do this sort of don't care feature extraction approach. Okay, and then ultimately what I want to do with this is take all of this approach and package it into a way that a biologist can grok and get something out of their data. And so I'll skip over the details of getting. What I'm doing is for every image, I'm computing the 6 nearest neighbors. You can do any number of neighbors that you want, so that a biologist can do. I'll skip all this. All right. So this is kind of what I see the project going towards. So this is a completely static thing, but it should be dynamic. So every point here, this is the PCA decomposition of the data. Every point is an image or a gene that's been disrupted. So here I've highlighted in orange a particular image that the researcher might be interested in. In blue are all the neighbors. So these are the images most similar to your image that you're looking at. So lots of genes are being done in replicate. These are the replicates. So you can see that they look more or less the same. So if your replicates don't look like your original one, then probably don't want to be looking at that gene because there's something fishy going on. And these two panels are cross-sections of that 2D matrix of images and features. So for this particular image, this is the feature profile. So each of these points corresponds to a particular feature. And for this particular feature, this is the histogram of what all of the different images have for that feature. So the way I envision a biologist using this is, OK, well, take me to the gene I'm interested in. OK, well, that has some middling value for this feature. What does it look like if we go to the either extreme of this distribution? And so very quickly, using all these pre-computed structures like your clusters and your nearest neighbors, a biologist can get an idea of what parts of their data are interesting and what the genes are doing. I'm not even going to go into this. Oh my God, it actually worked. OK, so if you haven't heard of mpl.d3, it directly converts matplotlib to D3 data. But these aren't linked right now, but you can do stuff like zooming into your plots and panning. So the main point of that is you can build something that's a nice interface using all of these frameworks. OK, conclusions and future directions. So I'll just go over this. Apologies for all the text. So as I mentioned, the number one thing, image clustering in Python is pretty close to out of the box. You use some standard libraries, in my case, scikit-image, scikit-learn. Combine them in a simple way, and you get some image clustering, which stuff like Google image search and whatever work on. I think the interactive GUI idea is feasible. All of these search functions are very fast, so that's the main goal. The goal is not an easy GUI for the simple one-dimensional feature analysis that most screens do. There's actually excellent software called Cell Profiler that's also built using the scientific Python stack that does that. But the goal is to be able to define a feature set quickly and easily in Python and ship interactive multivariate exploratory analysis to the biologist. One really interesting application for this work is to use the clustering and the neighbors network for functional prediction. So if you have a set of genes that you know are involved in this biological process, and you have some new gene that you hadn't heard of, and it's right next to all of these in terms of its effect on the cells, then you can be pretty sure that that gene is involved in that process as well. So suddenly you've got new biological knowledge that didn't exist before. And some minor improvements. So I've called it Husk, and when I did that, it was just because, as I said, this is live. Which is high-content screening unsupervised sample clustering. Because it's short, pronounceable, and it's not a word, but it's not actually Googleable. There's tons of things called Husk. I want to improve the image preprocessing. So I showed you some clusters that had some pretty good images, but some of them have massive artifacts. So it'd be good to get all the images to a nice, clean state before doing the work. And as I said, k-means might not be the ideal choice for this. So if anyone has experience in all of this stuff, and in particular in interactive data analysis, I'd be really interested to talk to you. And thanks for listening. Any questions?
Thanks very much, Juan. So we have quite a lot of time for questions now. Do we have any questions? Yeah. Okay, yeah. So there's been a call for Chris Lear. Is Chris Lear here? Yeah, can you please go to the registration desks? It's a call for you. Okay. Yes.
Thanks, Juan. That was a good talk. Just a quick question. What is the size of the data sets that you're dealing with here?
Right. So the data set that you saw is a very tiny subset. It was 1,700 images. The full data set for this screen is 45,000 images. The full set for new screens is 500,000 images. And this is after doing merging. So a lot of the images are actually taken as mosaics. And then, so after you merge them all, you get this size of data. So in the new sets, they're 500,000 fairly big images. A decent size.
Any other questions? Yep, I have a question. Yeah, Juan, would you be willing to share any of the future goals for the Scikit image project? Where do you see it going in the next few years?
Okay, so the thing with Scikit image is that it's kind of scrappy, and I mean, it was really interesting. We came out with the paper just recently, and as we were all submitting figures to it, like, wow, that's what you do. That's really cool. And completely different fields. So everyone has a bit of their own agenda, and so long as the features are seen as valuable by everyone, then they go in. My agenda is biology, so I want to make it easy to get support for common biological image formats, support for 3D data, which is right now pretty good, but not excellent. There's a lot of speed improvements to do. We have registration that is currently non-existent, so trying to align it to images that mesh together. Fancier image segmentation, revamping the I/O. Does anyone have any particular image wish lists? Yeah.
Yeah, I saw a job ad quite recently from CSIRO about phenotyping of whole plants. Within some field, you've got some number of actual plants. You grow the plant to a not necessarily mature size, but to a size. So you're not just phenotyping cells; you're phenotyping whole organisms.
Yep.
So is software?
Then you take a picture. Is that what's happening, or?
Sorry, what was the question?
Once it's grown, you take a picture of it, or are you doing?
Yeah, or you take video of it, or you do multispectral imaging of it, or you do something having to do with computer vision. So
Yep.
does it sound? It sounds to me like software like your sort of clustering software would be useful for identification tasks, even in that area.
I think so. So this project is very much in its infancy. It's sort of been me half-time, less than half-time for a year. But I think this project, part of the design of it is to very easily define new features. So long as you can extract relevant information out of your images or videos, or even if people are going in and measuring various aspects of each plant specimen, then yeah, I think this framework would be really useful. Again, because when you see something once, it could be a fluke, and when you cluster it and see it happening multiple times, then you're more confident that you're onto something.
Hey, so far there seems to be a common theme in the talks, and it involves programmers providing methods for wet lab or more scientists to get the data. Do you have any comments on the Python stack, how that's developing or growing to support easy application development to push data to the scientists?
I think Python is close to as good as it gets in terms of ease of use and finding tools that already exist to do whatever you want. And also, there's a huge education effort going on with software carpentry, and that's usually or mostly based on Python for the reasons that I said before. I should mention I actually trained as a biologist and slowly picked this up, and now I'm core developer for Scikit-Image. So I think that just speaks to how easy the ecosystem is.
Yeah, hi. I apologize my ignorance about Scikit-Image. To develop the feature vector, is there anything within there to kind of automatically extract features from images in order to enable clustering, or is it kind of every single domain needs to do manual feature extraction? There's
Nothing completely generic in there, but that actually might be something that we might be interested in. Yeah, so I had to develop. There's a lot of excellent primitives for it, which is how I worked here. Nothing out of the box, but that's a possibility if you want to submit. Like, it doesn't have to be a pull request. It can be raise an issue on GitHub. I think